Ka tangi te titi, ka tangi te kaka, ka tangi hoki kua hau. Tihei mauri ora. E ngā iwi o ngā haue whā, e ngā karangaranga maha, e ngā manuhiri tuarangi, tēnā koutou, tēnā koutou, tēnā koutou. Tēnā koutou i o tātou tini mate, hoki atu ki te kaitiaki o te pō. Haere, haere, haere atu rā. Te hunga mate ki te hunga mate, te hunga ora ki te hunga ora. E ngā manuhiri, nau mai, haere mai. Haere mai e raro i te korowai o te rangi Mare e. Ka nui te koa me te hari ki te kite i a koutou. Tēnā koutou ki a koutou kua tae mai nei ki te tautoko te kaupapa o tēnei pō. E hara ahau i te tangata mōhio ki te kōrero o te rā e tika ana kia mihi atu i te wā nei. Nō reira, tēnā koutou, tēnā koutou, tēnā koutou katoa. We will now start this evening with a karakia. Me inoi tātou. Tūtaua mai i runga, tūtaua mai i raro. Tūtaua mai i waho, tūtaua mai i roto. Kia tau ai te mauri tū, te mauri ora ki te katoa. Haumi e, hui e, tae ki e. It is my great privilege to have been invited to open Professor Richard Mithen's inaugural lecture this evening and to welcome you all here. Apologies that I was not able to join you in person. Welcome also to our guests joining us by Zoom. I first met Richard a couple of years ago when he first moved to New Zealand. In one of our early conversations, he mentioned, just casually, that he had been spending his time reading and learning about the history of Aotearoa, about our cultures, and about the Treaty o Waitangi and what the treaty means for those of us who live here. That really struck me as such a respectful way to begin one's journey in another country and a real acknowledgement to the people of this whenua. Since then, it's been my pleasure to be part of Richard's team here, well, kei reira, at the Liggins Institute, and to work with him as part of the High Value Nutrition National Science Challenge. In my time getting to know Richard, his passion and enthusiasm for the native plants and bird life of Aotearoa has opened my eyes to the amazing biodiversity that we have here and which it is our responsibility to protect. On that note, I'm really excited to hear Richard's presentation, so I'll hand over to Professor Frank Bloomfield to continue getting the evening underway. Kia ora.
Kia ora, Mika. E ngā mana, e ngā haue whā, tēnā koutou katoa. Nō mai, haere mai, e ko ana tēnā kou kua tai mai koutou, ki te whakanui i te kaupapa o te rā. Nō reira, e aku rangatira, tēnā koutou, tēnā koutou, tēnā koutou katoa. Thank you, Honorary Associate Professor Mika Foster, for that mihi and karakia, and welcome to all of you, both in person and in Zoom, to this inaugural lecture this evening. I'm Frank Bloomfield, the Director of the Liggins Institute. I'd particularly like to welcome Richard's family who have joined us by Zoom, Pat, Claire, Stephen, David, Rowan, and Laura. So an inaugural lecture provides a way to celebrate and recognise the mark of distinction conferred by a university when it awards the title of Professor, the highest academic rank and an achievement well worth celebrating. Professor Mithen has chosen to deliver his lecture in academic regalia. This mode of apparel for academics dates back to the 12th century, when scholars were all members of religious orders, and the clapperclouser, or the gown, is a development of the priestly pluvial. The hoods at that time covered the tonsures' heads and no doubt kept the monks warm. In the 14th century, the statutes of some English colleges forbade excess apparel and prescribed the wearing of long gowns, many of which used to be closed, and they still are for many American universities, to prevent the individual show of excessive finery. Richard's, as you can see, is open, so you get the full glory of his tie this evening. Specific details of academic dress were introduced around the time of Henry VIII at the universities of Cambridge and Oxford, and most regalia in New Zealand are based on those specific colours. but the current convention of specific colours for different degrees is a relatively recent invention. Richard's gown is for a PhD at the University of East Anglia, and my one is a PhD from the University of Auckland. As you heard from Associate Professor Micah Foster, Professor Mithen joined the Institute in January 2009 from Quadram Institute Biosciences in Norwich, the UK. But a number of circumstances that I don't need to elaborate for you have meant that this is the first time we've been able to recognise his appointment as a professor to the University of Auckland. Richard started his academic career studying plant science at the University of Wales, graduating with a first-class honours, followed by a PhD at the University of East Anglia. After 4 years in Africa, he returned to Norwich, where he spent most of his career before moving to Auckland. Richard was actually first appointed to professor at the University of East Anglia in 2000, actually 20 years ago this month, and that means that he was an exceptionally young professor indeed. And he was also a professor at the University of Nottingham before returning to Norwich, so this in fact is not his first ever inaugural lecture, which is why he's looking so calm and relaxed. His career at the Institute of Food Research, which later formed a key part of Quadram Institute Biosciences in a joint venture with the University of East Anglia and with the Norfolk and Norwich University Hospital, was impressive to say the least. From research leader to programme leader and then acting director of the Institute of Food Research, a role in which he was instrumental in the formation of Quadram Institute Biosciences, Professor Mithen's academic achievements are substantial. He's published over 100 papers with an H-index, which is a commonly cited metric of impact of research, of 46. One can argue about the merits of these different metrics relating to research publications, but what that number means is that almost half of all Richard's publications have been cited by others at least 46 times, and many of them hundreds of times. And this tells us that when Professor Mithen writes a paper, it is one that has something to say and that others want to know about and to use in their own research. He doesn't write papers that, once published, disappear into the graveyard of publications never to be heard of again. Professor Mithen also has filed 11 patents, at least 8 of which relate to health benefits of foods, again demonstrating both the impact and the innovation of his research. He has always demonstrated a keen interest in students and in the careers of early career researchers. He was a member of the Doctoral Training Programme Board for the Norwich Research Park and also had the role of Director of Graduate Studies for the Institute of Food Research from 2010 to 2015. And Professor Mithen has been the primary academic supervisor for over 35 PhDs, which is a substantial achievement. So as you can tell from Professor Mithen's lecture title, his career has spanned continents, and it's also spanned the breadth of research, from plant collection, genetic breeding, through identification and quantification of specific chemicals in plants, to testing those chemicals in randomized controlled trials, and then to spinning out his own company, the Smarter Food Company, of which he is founder and chief scientist. Professor Mithen's background therefore places him ideally for the role he has taken up here in Aotearoa, New Zealand, as Professor of Nutrition at the Liggins Institute and Chief Scientist of High Value Nutrition, Ngā Kai Whai Paenga, responsible for leading and directing the science strategy and quality of one of New Zealand's national science challenges, with the goal of transforming the benefits to New Zealand from its food and beverages through excellent science focused on health benefits. So Richard, I'd like you to come and tell us all about tall nettles from plant collecting in Africa to nutrition research in New Zealand. Professor Richard Mithen.
Ngā mihi nui ki a korua. Ko Mika, ko Frank, mō ngā kupu whakatū. So thank you, Mika, and thank you, Frank, for that very nice introduction. And before I start, I've got 3 comments I want to make. And firstly, and probably most importantly, is to really thank the University of Auckland for this considerable honour of being appointed professor at this very great university. It's not something I've accepted lightly, and I hope that my work here will do justice to this great institution. Secondly, I wish to dedicate this next 50 minutes or an hour to 3 people very dear to me. My son, Rowan, my daughter, Laura, and my partner, Claire. And I think that they are zooming in. And they were all expecting to spend a substantial part of this year and indeed last year in New Zealand, but of course events have meant otherwise. And so you haven't had a chance to meet them. So let me just tell you a little bit about them before I start. Rowan is in the middle of a medical degree. He's training at the moment on the wards at North Tees Hospital in the UK, a COVID hotspot. He's a passionate surfer and a pretty good chess player. Not quite as good as his father, of course, but you know. And I should note this recent run of wins is more luck than anything. My daughter Laura is coming to the end of a degree in ecology and conservation, and largely online as it is in these days. And she's not a surfer, she's a passionate gardener. This isn't a photograph of her garden. I can immediately see that because she hasn't quite worked out the skill of growing things in lines. Her gardening is more patchy. But she's an absolute fungus fanatic. And at the moment, she's spending a lot of time growing a whole range of different edible fungi in the different ways. And Claire is also finishing a degree in history at the University of East Anglia. And that's great, actually, because it means my days in Auckland, where I discuss the merits of eating kiwi fruit or kumara or milk or meat, are sandwiched between discussions of African American internationalism and the history of Art Deco architecture, both things of which Claire is very passionate about, but probably not as passionate as she is about German Shepherd dogs. And that's something that I'm still working on, but I'm getting there slowly. And indeed, this particular dog will focus a little bit later on in this talk, because that dog is all part of my journey to New Zealand. And before I start, I'd like to say a big welcome to other members of my family who I think are zooming in from the UK, despite it being rather early in the UK. It's about 6 o'clock in the morning here. And one of the ironies of COVID is that, you know, we've all been social distancing. And indeed, you could say I've taken that somewhat to extremes. But I've talked to many people, and it's true in my family as well, that our embrace of a number of technologies, such as Zoom, has meant it's brought a number of families together. And for that, I'm most grateful. And here you can see my immediate family, and there's my eldest brother, if I can get this working, eldest brother up there, David, with his wife, Sue. There's my brother, Stephen. He's my twin brother with his partner, Sue. Another brother of mine with his partner, Monique. My sister with her partner, Steve. There's Claire up there all by herself. And of course, in the middle there's my mother, a kind of matriarch of the family, mother of 5, grandmother of 12, and great-grandmother to date of 8, although it will shortly be 9, and no doubt before long it will be 10. So welcome and good morning to you. So let me start, and I thought I'd give this lecture a title. It's not something I often do. And I thought I'd call it Tall Nettles. Now, a number of people thought I was alluding to this tall poppy syndrome, which is in this part of the world. And I have to admit, I wasn't quite sure what that was and looked it up. And indeed, that would have been really clever, but I'm afraid that wasn't the case. The Tall Nettles is the name of a favourite poem of mine. And it's been a favourite poem of mine since I was in my teenage years. And like all great works of literature, it can work at different levels. And at one level, it's a rather pleasant little poem about an overgrown farmyard. And at another level, it says something very profound about who we are and where we're going, at least it does to me. And I'll end this lecture today by sharing that poem with you, and you can see what it says to you. Now, that poem was written by a poet called Edward Thomas, and he was mainly a book reviewer, but he wrote poetry just for 2 years before he died at the Battle of Arras in France in 1917, at the age of 39. And his great friend was Robert Frost. Now, Robert Frost, of course, wrote the poem The Road Not Taken, and some people say that's the most famous poem in the world, and indeed it was used in New Zealand some years ago to sell Ford motor cars. And that poem alludes to the different paths one may take in one's life. Do you go this way or do you go that way? And what that poem says, if you go that way, despite what you might think, you can never go back. And that seems to be a good poem to use an occasion like this, because any academic career, and even maybe any career, is all about making those choices. And as I tell you my academic journey, you'll see that along that route I had a number of choices to take. But I like tall nettles. And one reason I like tall nettles is that it speaks to me of the countryside of southern England, where I spent much of my formative years of my adolescence amongst the hazel coppice with its carpet of ramsons, and the bluebell, the beechwoods with its swaths of bluebells. And it was kind of here that this academic journey starts, I'm going to tell you about. And I went to a fairly traditional boys' school with my brother Stephen, where not only we did science, but we did Latin and we did Shakespeare. So I set myself a little task of getting a little bit of Latin and Shakespeare into this lecture, just to demonstrate to you what a well-rounded education I had. And I left here to go to university, and I suppose, in retrospect, against the expectation of some of my teachers who thought I'd go off and do something like medicine or something, I chose to go to university in North Wales to study marine biology. And at that time, I was very interested in farming seaweeds and eating seaweeds, and I thought this would solve many of the impending problems of the world, and I may still feel that. And it's quite ironic that here at a later stage in my career, I find myself engaged in this kind of research. But I went to university there, but I didn't actually stay studying seaweeds very long, I'm afraid. I think it was a matter of weeks, or possibly even days. And I got rather more enamored with theoretical ecology, population biology, and mathematical modelling. And I taught myself to code, and I began making mathematical models, predator-prey interactions, which were quite fun, and I seemed to be able to reinvent for myself some of the theoretical models that other people had done. And as my models became more complex, they became more chaotic. And this indeed was quite fashionable at the time, and intuitively we feel that as ecological communities become more complex, they become more stable, but actually if you do modelling, they become more chaotic. And this was quite interesting to me, and I had a great opportunity, it was a great university, in my third year as undergraduate to do my first real research experiment. And that experiment involved growing a big flat area of this plant, Lapsana communis. It's a kind of bit of a weedy plant, but it has found its way to New Zealand, and the little photograph at the bottom was a picture I took of it at Cox's Reserve on one of my lockdown walks a few weeks ago. And I grew this really flat, beautiful lawn of this plant. And then one night, catastrophe struck. There was a kind of leak in the roof of the glass house, and I came in the morning and it was all patchy, hopeless, disaster. And I took two things from that, which I think has stayed with me throughout my research career, possibly throughout my life. The first was that stuff happens. And indeed, it really has happened in the last couple of years. But secondly, you need to make the best of it. You need to make the best of it. And I think I did make the best of it. I published my very first academic published paper from that study back in 1984, leading to an 18 index of 57, not 46. And it was great. But I took something else from that as well, in that while I could model chaos, you can't model catastrophe. And indeed, it seems a bit of a modelling catastrophe seems a bit of an oxymoron to me. And so instead of going off to do a PhD in theoretical ecology or such like thing, and I was engaged with Robert May at that point at Oxford, I took myself off to work on some farms, which was probably a fairly sensible thing to do. And then I borrowed some money from my mother, and I took myself off to Africa. And I spent the best part of about 8 months travelling in southern Africa. I did a bit of work at a research station. And it was a great time. Not time to tell you about that, unfortunately, today. I haven't forgotten, by the way, Mum, if you're listening, that I still owe you a little bit of money. I'll go on to that. And I came back from Africa after a year, and I started to do a PhD at the University of East Anglia in a solid kind of subject, plant pathology. And again, I actually had some interest in fungus as well. And this was great. And I got on really well with my supervisor, Dr. Brian Lewis, and I'm still in touch with him. I saw him a year ago. Great guy. And I really made really good friends there with the other people in the lab at the same time. Philip and Fiona and Martin, and then his partner, Helen. And they are still my very good friends. And I think that's often the people you meet doing a PhD, you stay with them throughout your life. But there was a slight issue in that I don't think I was as engaged in that PhD research as maybe I could have been, and certainly not as engaged as I like my students to be. And I found out through writing letters, this was the days before email, days before the World Wide Web, I'm not sure there was a fax machine in the department at that time or not, that there was an international program to collect and conserve wild brassica species which grew on the cliffs around the Mediterranean. And I thought, oh, this sounds great, actually. So I wrote letters, and eventually they wrote back and they said, yes, you're welcome to come along, but you need to pay your way. Now, of course, this was a bit of an issue. I was already in debt. And before I come on to that, I just need to, a little diverge, tell you a little bit about the university and where it is in England on the Norwich Research Park. And this is the Norwich Research Park. It's a very big concentration of life science institutes. The university sits back there. Now, whether the university is part of the Norwich Research Park or adjacent to the Norwich Research Park is a fact that many vice-chancellors, pro-vice-chancellors, directors spend hours and hours discussing. It doesn't really matter to me. And I was actually going to spend a large part of my research career, I didn't know at that time, at this research park. Firstly at UEA, more latterly at the John Innes Centre, then at the Institute of Food Research, and then at the Quadram Institute. And during that time, I formed very strong collaborations with the Genome Centre and the University Hospital, and I'll come on all to that presently. But the point of this little divergence here is to tell you that in the 1980s, the director of the John Innes was Professor Harold Woolhouse. Now, you need to think of Harold Woolhouse as a kind of Frank Bloomfield kind of figure. He's a director, very important person. And even at that age, I knew what directors did. And they sat behind big desks in very nice offices, and Frank's got the windows. And in those big desks, they have big drawers. In those drawers, they have bags full of money. And that's what directors are about. And so as a first-year PhD student, I went over and I just wandered into Harold Woolhouse's office. You could do that in those days. In these days, you probably have to make an appointment 3 months in advance and be security vetted. But I just wandered in, I explained to Professor Harold Woolhouse I was a first-year PhD student, and could I have some money, please, to go collecting plants on the cliffs of Italy? And he said yes. And then his next question was, how much do you want? But I hadn't really got that far of thinking that, and I asked for some pitiful small amount, which he readily agreed to. And it was actually I was asked that question another time in my research career, and I was equally unprepared for that question. And then just as I was about to go, Harold said, there's one condition. Bring me back some seeds of this plant, Moracandia arvensis. And I said, Professor Woolhouse, nothing will give me greater pleasure. But of course, I'd never ever heard of this plant. I had no idea where it grew or anything about it, but I readily agreed to do that. And so a few weeks later, I found myself sitting on the seawall in Bari, in southern Italy, and there I'm at the end there, in the company of three very important professors. There's Professor Pietro Perini from Italy, if I get this to work, here. Professor Mats Gustafsson from Sweden, and Professor Gomez Cesar Campo from Spain. And we had a wonderful time, and my goodness, how kind they were to me. And we went collecting down the Amalfi Coast. Now this is a nice picture. It's rather untypical because it's a bit of an untypical habitat. The habitat for these plants were more like this on these cliff faces, and if you can see just down there, there's some flowering wild brassica, and a lot of this time involved climbing up and down these cliffs. I only fell off once, but it was quite a long fall. But of course, we weren't collecting flowers, we were collecting seeds like this, and we collected from down this bit of coast here, the Amalfi Coast, onto Ischia and Capri, and then around Sicily. And one of the plants that I collected right back in the early 80s was this one, Brassica villosa from Sicily, and I'm still working on that plant now, or the genes in that plant. Now, when I have a habit, and I rather indulge in this in New Zealand as well, about when I'm walking around, I tend to taste the plants I'm seeing, nibble at them, and presumably at some point this will poison me. And I tasted these brassica plants as I went around, and it was really interesting because many of them had quite a hot taste. And this is because when I bit into them, I was causing a chemical reaction of this compound here, and it was cleaving here by an enzyme and releasing a mustard and isotonic. And you're familiar with these compounds, because if you eat sushi and put a little bit of wasabi on it, this is what you're tasting. And if you eat things like watercress, that's what you taste. And they have a hot taste. And they have a hot taste because that compound stimulates in your mouth your TRVP1 receptor, which is a receptor which senses temperature. And it's quite interesting that these plant chemicals interact with the receptor and they taste hot. And it was interesting because they didn't all taste the same. Some of them tasted very hot, some of them were quite mild. And then as the weeks went on, I realized that there were much more subtle changes. And over the years, I realized that these subtle changes were due to chemical changes in this side chain here, rather subtle changes, but have an influence on flavor. And that's the difference between if you eat bok choy or watercress or broccoli. They all taste a little bit different. And again, at that time, I didn't know that I was going to spend much of my career working these chemicals. However, I went back to Norwich. I did my PhD in this area, began thinking a little bit about how you could use plant genetics to explore biochemistry. But out of the blue, when I finished my PhD, I was offered a position in Africa as a plant collector. How weird. My goodness. So, and what was this about? And this came out of the blue, this job really, and it was with an international organisation, one of the CGIAR centres. At that time it was called the International Board for Plant Genetic Resources. It's now called Biodiversity. And this was about cowpeas or black-eyed beans, and you may be familiar with these. You can buy them in shops in New Zealand. It's a very important subsistence crop in Africa. and they suffer from a lot of problems, pests, diseases, and the nutritional profile isn't what it may be. And so there's a lot of interest in collecting the wild relatives of these to be using in breeding programs. And I suppose even at that young age, I had a bit of experience of Africa, a bit of experience of plant collecting, two kind of came together a little bit. But before I was offered that post, there had been quite a big program, a kind of 3-year program, which had really looked through tens of thousands of herbarium specimens in places like Kew. And this is the Kew Herbarium. And it's an absolute wonderful place to spend time. If you look through those cupboards, you find specimens collected not just by Banks, but by people like Darwin and such like. And it's absolutely fantastic. And that long study, not just at Kew, but in Missouri, Berlin, Paris, had clearly shown that the wild relatives of the cowpea were not to be found in West Africa or East Africa, which is what the book said, but were actually in Southern Africa. And so after a few weeks in Rome, I found myself in Zimbabwe, working jointly between the National Herbarium and Botanic Gardens and the University of Zimbabwe, with quite a big budget. And my task was to work with national programs in those southern African countries to collect and conserve the wild relatives of cowpea. Now, I've mentioned already Harold Woolhouse, who's been quite influential in my career, and I'd like to mention a few other people as go along, and one of them is Bob Drummond. He was a keeper of the herbarium, a man who really gave me a huge amount of help. He was also, I should say, a man of certain eccentricities. He had a great passion for his science, and he's passed away now, and sorely missed by many. Now, let me tell you a little bit about Africa at this time. And again, you need to think this is in the late 1980s. There was an apartheid regime in South Africa, which meant that officially I couldn't go to South Africa. But for practical reasons, I had to a couple of times, and I was fortunate to be able to travel on a second passport. And one of the reasons was to collect a Land Rover, which had been imported into Durban, and to drive it back to Zimbabwe. And that was quite an experience. The only thing I'd driven up to that time was a rather falling apart 2CV. But anyway, and then also there was a rather nasty civil war going on in Mozambique and in Angola. And Namibia was largely out of bounds as well because of South Africa fighting their war from their borders. But the countries of Botswana, Zimbabwe, Zambia, and Malawi were peaceful, and at that time, Zimbabwe was a wonderful place to live. It had a booming economy. There was a policy of reconciliation. It was a wonderful place, and I very much valued my time there. Let me tell you a little bit about what I was doing and the places I went, and then I'll share with you 3 short anecdotes. I only went to Mozambique once. And I remember flying from Zimbabwe very high over the country, fly very high so you don't get shot down. And then over Maputo, the plane spiraled down and down and down, almost seemingly forever. And in Maputo, kind of war-torn Maputo, which is right down here, I met up with some amazing students from the university and academics from the university who, despite the war, were continuing their academic endeavours. And we went and collected plants and other biological specimens on offshore islands in the Indian Ocean, including Vigna marina, which was the Vigna species I was after, which grew on those sands. And that was an amazing experience and such a privilege. I spent quite a long time, or many visits, to the Eastern Highlands of Zimbabwe. And here, here's the Chimanimani Mountains, and the Chimanimani Mountains are here on the border of Mozambique, and they are very beautiful. And you spend a day driving down from Harare, and the mountains face west and have a lot of quartzite in, so they kind of shimmer in the evening sun. And a day climbing up those mountains, and on top there's big grassy plateaus, and they're we were collecting, or I was collecting, a very rare species, Vigna nervosa, which we found several times. Going further north to Zambia, with these great expanses of Miombo woodland, and I spent some time up in the copper belt here trying to get the more tropical species. It was actually a pretty difficult place to be. The price of copper had fallen a lot over the previous 20 years, and so it was a very run-down area. But here, this is the bridge over the Luanga River on the way to Malawi. And I have very fond memories of coming up to that bridge. And as was typical at that time, there are roadblocks and young teenage soldiers with AK-47s in one hand and the beers in the other hand. And you always feel a bit anxious in those situations, as you would. And I remember that within 10, 15 minutes, really chatting about Manchester United and football, a subject of which I have relatively little interest, but a subject you should always know a little bit about, because it can get you out of any kind of situation. Maybe not in New Zealand, though, which doesn't seem to work in this country. But anyway, from Zambia, you go on to Malawi, and I had a wonderful long time in Malawi, collecting plants right from the mountains in the south, Mount Mulanje, Mount Zomba, including a time I got very ill, actually, with amoebic dysentery in Zomba. And then collecting right the way up the shores of Lake Malawi to the Tanzania border, collecting a species called Vigna luteola and another legume called Sophocarpus lunatus. And I remember in particular a wonderful Christmas I spent without my national counterparts up on the Nika Plateau. Very special place. And then back to Zimbabwe. This is Zambezi Valley, the Rift Valley. Very low-lying, very hot. Not actually very good for plant collecting, not the plants I wanted, but very good for large wild animals. And I spent quite a bit of time down there. I was fortunate to do some teaching on some camps for kids from rather deprived schools, and had the opportunity to see lots of and many close encounters with the large wildlife of Africa, including elephants and lions and hippopotamuses and rhinos and such like, wild dogs, and in particular one very close encounter with a leopard, which I can tell you about later if you wish. And then the Okavango, that great, fantastic inland delta in northwest Botswana. And just an amazing place. I think I regret not going right deep into the delta. It was difficult to justify that from work, but I certainly collected on the fringes of that. But my very favourite place was undoubtedly the Kalahari Desert. And I was fortunate enough to do a number of collecting trips through the Kalahari, and I'll tell you about one of them shortly. And so before I move on, I'll tell you 3 short anecdotes, 2 of which may be slightly amusing. This is Mountain Nyanga. This is in the eastern highlands of Zimbabwe, slightly north of the Chimani Mountains. And this is the sacred mountain of the Shona people who live in this part of the world, and is a place to be treated with considerable respect. And it was on the slopes of this mountain I found a totally new species, within the genus I was working on, Vigna. And so I had the opportunity to describe and name the species, and I called it Vigna inyangensis, the Vigna of Inyanga, in respect for this place. And here, finally I understood why I did schoolboy Latin, because it allowed me to do that Latin description, which I very much enjoyed. And I would also just show that these lovely line drawings of Vigna inyangensis were done by my friend and companion for some of these journeys, Helen, who was a very talented artist and would go on later to be the mother of my children. The most amazing collecting trip I went on, though, was to the Kalahari. And this year there had been more rain than in the previous 80 years. And we left Harare, pouring with rain, and we drove on the tar road down to Herbarium. In Herbarium we met our local counterpart, a very talented botanist called Ellen Clarkey. Ellen was great. She was later to die in a car accident. And almost as we drove into Herbarium, the sun came out. And within a week, we set off across the desert. And it was really like a David Attenborough documentary. You know, the desert just bloomed around us. And we went right across the central Kalahari, over to the Okavango Delta, and then down the border with Namibia, across this very dry area of the Kalahari, down to the South African border and back, and it took several weeks, and it was a most extraordinary collecting trip. And Bob Drummond paid me, I think, probably one of my greatest compliments by saying it was the second best plant collecting trip he'd ever been on. And his best collecting was right back in the 1950s in East Africa, but that was a great compliment from Bob. One of the interesting plants we collected on this trip was a wild form of cowpea, right from the very driest parts. And I was particularly pleased with this because I'd seen herbarium specimens collected over 100 years ago in Kew. And everybody was rather doubtful we'd find these, but we found them in abundance. And again, we described these not as a new species, but as a new subspecies. And this was particularly interesting. Now, I did probably have one of my most remarkable experiences of my life right in the middle of the Kalahari, and I'll tell you about this. It doesn't have a big science content, but it may amuse you. At night, we would sleep around a campfire and look at that amazing southern sky at night. And indeed, someone asked me the other day, why did you come to New Zealand? And I said, I needed to see the night sky from the southern hemisphere once again. I've done that, actually, I've ticked that one off. But we were sleeping outside, and I think everybody was sleeping in tents apart from myself. I was sleeping on a stretcher, a camp bed around a fire. And I woke up in the middle of the night, and I felt this just huge weight sitting on my chest. And I mean, really heavy, and I couldn't talk and I couldn't move. And of course, this is what's called a sleep paralysis. It's the only time in my life it's affected me. And I was really there. And I remember lying there and going, trying to get this off me. And in my head, I knew that a great big snake had not only curled up and sitting on my chest, but had bitten me and I was going to die. And I really thought that very much. And I remember lying there thinking and looking at the sky. But of course I didn't. I woke up in the morning. And I said to one of my companions, I said, did you hear me call out in the night? They said, oh yeah, I think I did hear you call out. I didn't come out to see what was wrong. And anyway, it's quite interesting, and it kind of made me quite interested in this. And of course, in all cultures, you have these stories. And in the northern hemisphere, you have the Mare, it's called in this malicious entity. I don't know how malicious they are, but they come and sit on your chest at night, weigh you down. And of course, this is the origin of the word nightmare. Now, some weeks after, once I talked to this both people, they said, well, that's a witch. That's what witches do. They sit on you at night. And so I had this witch sitting on me in the middle of the Kalahari Desert. But there is a difference. In the northern hemisphere, witches fly on broomsticks. And in Africa, they ride on the backs of hyenas. And there were certainly lots of hyenas around at that time. Now, so that was quite an interesting experience, one that I haven't forgotten. And nor the next one, because excitement didn't only happen in the desert, it can happen in the suburbs. So let's go back to Harare before I move on and put some science into this talk. Here's Harare. Look, there's the Botanic Gardens, and there's the university. Oh, look, there's Avondale. I used to live in Avondale. It's a bit like Auckland, isn't it? And indeed, the architecture of Harare is very similar to Auckland. The downtown architecture, the suburbs, it's very similar. And when I first came to Auckland, I was rather taken by this. There are some differences, of course. In the 1980s, Harare had a ring of roadblocks around the city. And if you wanted to go out of the city, you'd have to talk to the army or the police to explain what you're doing. You can't imagine that would happen in Auckland in 2020. Now, when I wasn't driving my kind of macho Land Rover around Africa, which I rather enjoyed, and I must say I thought I was rather good at, I rode an old bicycle. And I often used to ride from where I lived in Avondale down through across this, what's called this flay. In Auckland, you might call it a reserve or domain or something. And it was a fairly big land, a bit wet, a bit muddy. And I used to cycle my bicycle across there. And one day I was cycling across there, and wow, I saw this species of plant I'd been looking for a couple of years by this time, Vigna monophylla. And it grew just a few hundred metres from where I was living, and I never saw this species again. A fairly widespread species through eastern South Africa, but very rare. I thought this is fantastic. So I made a note, and I went to the herbarium, and I came back, and I brought a map with me. And you had very good maps of Zimbabwe. They were paid for by the Scandinavian aid agencies, and I very carefully... I should say the plant was in flower, which there's lovely flowers on, but actually what I wanted was seed. And so I thought, I have to come back in about 3 weeks' time. And so I very carefully marked on this map some crosses exactly where this plant grew, so I'd know where to come back from, because by that time I learned that your memory isn't good enough. Now, what happened in the next week is that there was a bomb, and actually one of the houses fairly near where I lived was blown up. And of course, this is in the time of apartheid, and some South African insurgents had come up to Harare. They had positioned a mortar, and they'd fired that mortar, and they blew up this house, which was an ANC house. The ANC, of course, were fighting the freedom struggle in South Africa. Fortunately, nobody was hurt in this. And, you know, these things happen now and then. It wasn't that infrequent. I think while I was there was probably half a dozen explosions over a period of a few years. But anyway, so I didn't think much about it, but I went back to collect my seeds. And as soon as I got to the place, I was immediately surrounded by armies pointing guns at me and the police, and I was arrested for blowing up an ANC house. Because where I put my cross was almost the exact place that this mortar had been positioned, which blew up the house. And actually, I was at Mission Bay the other day talking to Ramya and Farha, and they said, what's the scariest thing you've ever done? They've been boasting about their bungee jumping, which is something I couldn't do. And I thought, well, I don't know if I did this, but it was a scary moment, because it did go through my head I might spend the next 30 years in a Zimbabwe prison. But anyway, I did get out of that one, but it did actually cast a shadow over the rest of my time there, because I was followed quite a lot since that time. And I remember one particular scary time of going to my office at the university and finding a little handwritten paper note on my desk saying, "We are watching you." And I have that note. Now, you may be getting the impression at this time in my talk that Africa was full of experiences, but maybe not much science. And I was kind of drifting around Africa a bit like a Victorian plant collector, while the kind of engines of molecular biology were being stoked up, and I was a little bit concerned I was missing out. So I thought after a time it was best to head back, and I head back, and fortunately Professor Woolhouse offered me a position as a research leader at the John Innes Centre. It wouldn't happen these days. These days you'd probably need 3 papers in Nature and before they even looked at you. But anyway, Harold was a great guy. And the John Innes was an amazing, wonderful place to work. Now, this isn't the current director. This is Professor William Bateson, who was the first director. And Bateson was actually the person who first coined the word genetics. And what you can see there is a letter where he says, let's call this new science genetics. And the John Innes has had a history of over 100 years doing fundamental work in genetics. They apply it to plant and microbial science, but has lots of applications to all areas of human genetics and medicine. And it was quite an amazing place to work. I stayed there for about 11 years, and then, as Frank said, I was offered the chair of crop science at the University of Nottingham. And so 20 years ago this month, I gave a previous inaugural lecture. And Nottingham was a great place. Many of you may have associations with it. This is at Sutton Bonington. And I need to mention Mare Tracker. And I'd been there not so long, and there was a knock on my door, and there was a student there, and Maria was doing a master's in plant science, and she said, "Oh, Professor Mithen, I hear you might have a PhD position available." I said, "Well, yeah." I said, "Well, let's go and have a chat, have a coffee." And halfway through this chat, she kind of stopped and she reached across and she said, "Richard," because by that time I told her to dispense with the formality. She said, "Are you a proper professor?" And maybe I hadn't fully embraced the kind of University of Nottingham professoriate at that stage. I'm not sure I've ever did, really. And then the other anecdote about Maria is that she started this PhD with me cloning a gene in Arabidopsis. But within, I think, 4 or 5 weeks, I was spending most of my time at the Queen's Medical Centre in the main part of the city, and Maria was finding out how to grow KK2 cells. And that very much marked the transition of my research career from being plant-based to really focusing on human nutrition. And after a few years at Nottingham, we both moved back to Norwich, where Maria now is a research leader in gut microbiome and doing a brilliant, absolutely brilliant job. I had a very nice Zoom call with her last night talking about science. And I led 3 fairly large program grants in food and health and worked increasingly with people at the hospital. And here you can see Mr. Robert Mills, a consultant urologist at the hospital. I've worked with him for many years. And next to him, Antonella. Antonella worked in my research group, again, for a long time, and is now oversees all human studies from the Quadroom. And Tracy Livingstone, who's one of my current students, and Tracy is a clinician who's just taken some time out of her training to do an MD degree, which has been slightly disrupted by COVID. And we have an intervention which is just about to finish. And as Frank said, in addition to the science at IFR, I was very involved with the transition of the Quadroom, of the Institute of Food Research to the Quadroom Institute. And this big building, and this building, I should note, goes quite a long way underground. And underground, you have the biggest colonoscopy unit in Europe. And the idea was to try and interface the clinical work with basic research in food and microbiology. And I think it's a great infrastructure, and hopefully that will come to fruition over the next few years. Moving on, and so over this period, and now I'm spanning actually a long time, about 25 years, looking back in retrospect, I really asked 3 questions. My first question, and again this was at the earlier part of my career, but it's continued actually today, was how do plants make complex metabolites? And I was focusing on glucosinolates, and I worked a lot on Brassica, but also on the model species Arabidopsis, the kind of Drosophila of the plant world. And make models of biochemical pathways is what you did, and then lots of genetic analysis. And it's amazing thinking back to those days where we didn't have any linkage maps, we began mapping Mendelian traits, QTL, and then, of course, the whole sequencing revolution. And the Journalists was a great place because they were pioneering genome sequencing of organisms. And more sophisticated models, and I just note one gene, MIB28, which I'll come back to. And then for many people there, the genetics was an end in itself. But for me, it was just a tool, because I wanted to use that genetics to really explore 2 interesting biological questions. One was plant herbivore and plant pathogen interactions. The second one is human nutrition. And indeed, these worlds have much to gain from interaction with each other, but they actually operate in largely completely separate spheres at the moment. And just briefly, the plant herbivore one was really great fun. This was mainly while I was at John Innes. And you can see here, if you increase the level of these compounds in plants, and these are increasing the level, you can get less damage by things like slugs and pigeons, but you get more damage by these specialized insects that have evolved mechanisms to detoxify or sequester these chemicals, just like some of the bugs do in our guts. And I had a particularly enjoyable time with working on more complex communities, and we showed that by altering the plant genetics, we could attract in a parasitic wasp, which would then run around and parasitize aphids on the leaves. So that was a great time. And then I also did some work, not in experimental populations, but in natural plant communities. And this is down on the South Dorset coast, and did some interesting kind of ecology, so it's population biology. And it was interesting for me just a few months ago to realise that this work had been picked up in New Zealand. And the models we developed at that time have been used to look at some population evolutionary ecology of some plants in the Southern Alps of New Zealand, which very much uses what we did there. But the main question I then focused on for the last, I suppose, 15 years, seems like 50 sometimes, was really it seems like an easy question, does eating broccoli keep us healthy? But it's an amazingly complex question, and food interventions are very challenging. And this is due to glucoraphanin. What drove this work was work that came out from the United States, from Johns Hopkins in the early 90s. And that work said that this compound sulforaphane, which is derived from the compound accumulated in broccoli, and the gut bugs in our gut generate this compound in our gut. This is a potent inducer of a transcription factor called NFR2. It does this by inducing oxidative stress. If you run around a block, you have the same effect, and what we need to do is to induce oxidative stress in our system, not consume antioxidants. And what does NFR-mediated transcription do? It switches on maybe up to 200 genes in different tissues, but mainly hepatic metabolism. And these genes are involved in a number of physiological processes which are probably thought to be beneficial. And you can see they involve things like phase 2 metabolism, antioxidant activity, and they modulate metabolism. And at the same time, there was also a lot of epidemiological studies coming out. So from the other end, and these said that people who ate diets rich in cruciferous vegetables, such as broccoli, were having lower incidence of cancer at different sites. And here you can see papers on prostate, gastrointestinal tract, and lung. And these two things seem to be, wow, that's interesting. You've got mechanistic at one level, epidemiological. So I kind of rather naively set myself the task of seeing if we could address this experimentally. And I thought, well, I know how to do this because I understand the genetics of these. And what you need to do is to develop broccoli with higher levels of these, and then you can see if you feed people these, whether they're better than people who have low levels. Naivety is sometimes exactly what you need as a scientist, and if you don't have that, you never do anything. So I used material that collected right back from when I did that collecting, when I was a PhD student, and integressed by conventional genetics, but a lot of DNA fingerprinting work, into a broccoli, and you can see by transferring this transcription factor allele called MIB28 into here, you increase the level about threefold. And then if you introduce a second copy, so you make it homozygous, it can go up even higher. And I thought this is great, this is how we can do it. And as Frank said, at that time, we also worked with a company to commercialize this broccoli, and it was on sale for quite a few years. The company that commercialized that then was bought by another company and such like. And so we took the intellectual property back, and at the moment we're developing that through our own spin-out called the Smarter Food Company, and that's going very well. So what did I do with this broccoli? And that we did a whole series of human intervention studies, and I'm not going to describe these all. And the first ones we really used broccoli itself, and the logistics is too complicated really. And these were really focused on what we'd now call kind of metabolic health. And my kind of take-home message in looking at all these studies, and of course we also did a lot of cell culture studies and a lot of seahorse kind of work. And it seems to me that, if you eat lots of broccoli or such, what you're doing is retuning your engine, and your engine being your mitochondria. And I think this is a very good analogy. And what it does, for those who are interested, it really helps fatty acid beta oxidation to be well integrated with the TCA cycle, and that avoids compounds like acylcarnitines accumulating in your plasma. And secondly, it really leads to citrate going down a catabolic route, going round the TCA cycle as opposed to being exported into lipid synthesis. And retuning the engine is quite a good analogy. If you do these studies on fairly young people whose engines work very well, you see absolutely no effect. Of course, if you do them on people such as me, who've now been around the block a few times, you can then see some improvement in their metabolism. But of course, you know, if your engine goes wrong, it doesn't matter how much broccoli you eat, it's not going to help. And of course, for all of us at some point, our engines will stop working altogether. And then the other series of studies they did was actually using a soup product. And these were really focused on prostate and prostate cancer in men. And here again, I'd like to just acknowledge two very important people in my career. Professor Colin Cooper, Professor of Cancer Genetics at the University of East Anglia. Very, very kind, very clever man. And also Jonathan Simons, who's the Chief Executive of the Prostate Cancer Foundation of the United States, who was very generous in their funding into my lab over many years. And I think they really decided to fund in food and health, an area which they'd never funded before, really being focused on drug interventions. This last study, ADAPT, isn't finished yet, and that's the one that Tracy's working on. And I'm not going to tell you much of the science here, but just the brief summary of the ESCAPE study. And this study was meant to take, 18 months, and it took, I think, 6 years to finish. It took an awful long time. And this was a study in which we recruited men who had early signs of prostate cancer. We took biopsy tissue from their prostate. We did RNA-seq, so we sequenced all the genes, we looked at their expression. And then we had them on a diet for a year. And then we took other biopsies from them, and we looked at what changes were happening in that prostate over that year. And you can see the red are the genes which have gone up, the blue are the genes that have gone down, the black are the genes which didn't change. And many of these changes were associated with the promotion of oncogenic pathways. So these people were at risk of much more serious cancer than they had. And we showed that using the high glucosinate broccoli, we could attenuate some of these changes. And in the very high one, we could almost reduce them very much. And that's work which, again, we're continuing with. And one of my ambitions in New Zealand is to undertake a much larger dietary intervention study here with clinical endpoints, which this one didn't have. Now, this is the Galloway Hills in Scotland. And some years ago, I was on a holiday with Claire. And it was a bit of a challenging holiday. We were in a camper van with a lot of rain, with a very large dog. But it was a lovely holiday. And at one point we saw, I said, look at that, Claire. Adventure before dementia. I said, we better write that down before we forget it. Claire didn't write it down, she took a little photograph of it. And so a couple of years after that, I think that stuck in our mind that maybe we had one more great adventure in our life to come. And so we decided to exchange the ornithological delights of North Norfolk, and here you can see the thousands of Brent geese which are now coming in at this time of year to overwinter on the marshes there. They'll be coming in from Svalbard and Greenland and Scandinavia, to the undoubted and remarkable ornithological delights of New Zealand. And here you can see one of them, and indeed this is my Shakespeare moment. And to paraphrase Miranda, when she opens her eyes and sees Ferdinand for the very first time, the first man she's ever seen apart from her father, the magician Prospero. Oh, brave new world that has such creatures in it. And indeed, what creatures you do have in Aotearoa, New Zealand. They extend far beyond the Kiwi, I should note. So that's brilliant, but there are many delights of coming to University of Auckland and the Liggins, and one of them certainly is working with this group of people. Joanne, Eleanor, Simone and Christine, and it's very nice that I can see Eleanor and Simone here. And I think we're a great team. I spend half my time working in the High Value Nutrition National Science Challenge. And I must say, I think at the moment we're really motoring and the program is really developing. And it's nice to be doing good science, but it's also nice to work with such a great group of people. And so thank you very much for that. And finally, I get to find out a little bit more about seaweed, right back so where I started. And then also, of course, I intend to get my own research up and running, which I'm now beginning to do. And of course, I'm very interested in this interface between plant chemistry and human health. But maybe not broccoli, maybe I've done enough broccoli, although we have got some broccoli work going on. And you can see here, these are the chemicals, very similar to broccoli, that you get from, this is wasabi, and also cress, and in New Zealand you have very interesting Lepidium species, Lepidium odoraceum, and you'd get these compounds. And these compounds, again, would interact with that TVRP1 receptor and induce NFR2 transcription, as does capsaicin from chili peppers. And then coming to Aotearoa, New Zealand, I was particularly intrigued by two of your endemic species, Pseudowinter colorata, Horapito here, and Piper excelsum kawa. Of course, these are taonga. They are really important species in Mare culture and to be treated with considerable respect and indeed reverence. And so it's been great that I've been able to develop such a great collaboration with Wakatu, a Mare-led company full of innovation, but a very, very values-led company at the top of the North Island. And that's been great, and that's allowed us at the Liggins to start exploring these plants and what health benefits they may produce. And of course, I'm fortunate in having another such great team, to be part of another really great team, and here to really thank and say what a pleasure it is working with Mika Foster, who you heard earlier, and you'll hear shortly, Chris Pook, Faha Ramzan, and Ramya Jakaparesh. And it's really great, and you know, I was thinking I had such a great meeting this morning with them, just talking about science and stuff, and it was great. And I kind of like to finish now and really saying what a pleasure it is to work with Ramya as a PhD student just going into a second year. And I was thinking actually some weeks ago about one's legacy as an academic scientist. I haven't finished yet, but I can think about this a little bit. And one's legacy may be one's papers one's published in more or less prestigious journals, and maybe more or less cited. Or one may have an institutional legacy, and I'm sure Frank will leave a legacy of what he's done at Liggins. And maybe I'll have a bit of a legacy at the Quadrium Institute. But I think one's greatest legacy, and probably one's most important legacy, is one's graduate students that have passed through your lab. And so I want to finish with saying thank you in two ways. And to thank all the graduate students I've had over the years, and to thank them for their contribution to my academic journey, of which I've told you a little bit about. But also to thank them very much for the privilege of being part of their journey, wherever that journey has taken them. So maybe I can just actually thank Joel and John, and Dorothea and Ben, and Karen and Michaela and Tracy, and Hugo and Claire, and Robert and Omar, and Danny and Ruth and Fatma and Dash and Maria, and Raki and Kathy, and Melanie and Golgi, and Lee and Philip, and then Claire, Thanasis, Peter, Amy, Rosie, and Jack. And those few at the end maybe need to just improve their social media profile a little bit. So I can get their pictures. And with that, I'll leave you with Tall Nettles, and you have a short amount of time to read that poem and see what it means to you.
Well, thank you, Richard, for an absolutely wonderful tour de force of a lecture. I think everyone would agree, taking us through your personal journey as well as an amazing scientific journey, I think showing the incredible breadth of work that you've done. You never did tell us whether you got those seeds from Mona Vale or not.
Yes, I did. I also got the seeds for Harold, and how good certain professors are. And Cesar Gomez Campo, when I was collecting, we did a 200-kilometre detour, especially to get those seeds for Professor Williams.
That's wonderful. Just remind everyone that there are reception upstairs, and finally, if you do need to get to see me, it doesn't take 3 months. You just have to get past Kate Summers. Now, I'm just going to try and get Mika Foster back on to close the day for us. I think.
My burning question, Richard, next time I see you is, what was that close encounter with a leopard? That sounded pretty intriguing too. That was a fascinating talk. When Richard was preparing this lecture, he described it to me as his version of a pepeha, so his way of introducing himself and establishing his identity and heritage and those things that are important to him. After listening to you talk e hoa, I think that was a really apt description, and what a fascinating journey you've had so far. Thank you for sharing. I look forward to the next instalment. So now, in closing this part of the evening, it is tika, or appropriate, that we finish with a karakia whakamutunga. Me inoi tātou. Unuhia, unuhia, unuhia ki te uru tapu nui, kia wātea, kia māma, te ngākau, te tinana, te wairua, i te ara takatū. Koia rā e rongo, e whakairia ake ki runga. Kia tina, tina, hui e, tai ki e. Tēnei ahau ka mihi atu ki a tātou, ka ti rā, pēnā koutou, pēnā koutou, pēnā koutou katoa. Have a lovely rest of your evening, everybody.